is laser. So, using laser confocal microscope, scanning microscope, you need to set up the wavelengths first. Lots of the time, 550 nanometer is for red, 625 nanometer is for green. Because similar to the fluorescence microscope, we do the Davis stain. All these like a different type of the like called cyto9 staining. We will know the live cell and data cell. We set up the wavelengths. So that's the same thing for fluorescence microscope. But what's special? Laser confocal scanning microscope, you could see 3D images. This is important. We not only to see the first surface, we will see the length, width, and how high it is. Why? Because laser confocal microscope, to do the observation of a very special bacteria called the bacteria biofilm. What's a biofilm will be forming? So I gave you a very easy example. Do you think this bench have a bacteria? Yes, obviously. And I tell you this bacteria compared to the bacteria growing in the tube in my lab is different. And this is biofilm. Now usually how they generate is you do a pull action of sanitizing and cleaning. So let's say a meat plant. Okay, we are processing those ground beef. It's very sticky. We did the whole day. And I'm tired, I worked like 9 hours. I want to go home. So they just spray the water. Okay, when you spray the water, the bacteria are going to grow. We will learn very soon. As long as you have a moisture, bacteria will grow. So the bacteria grow. But in a very poor nutrition environment compared to the media. So what happened? They will gradually to have a matrix generated. And this matrix is very heavy polysaccharides, which is sugar. Now, what's the bad thing for these biofilms? You keep lazy every day. The biofilm gonna grow very sick. And one day you say, okay, I get some time today, I'm going to clean it. It's very hard to clean. The reason is these biofilm, they're resistant to antimicrobials. So there is a research showing for normal bacteria, we talk about the bacteria in the broth, we call it a vegetative cell. If you are using chlorine solution, you may only need 50 parts per million, 50 ppm. If the biofilm, even you are using sometimes more than 1,000 ppm, the bacteria can still survive. Why? Because the biofilm have a matrix. The bacteria cell grabbed together, they have a chemical communication. We call it a chlorine sensing. And for gram-positive and gram-negative bacteria, there are different chemicals to guide them to resistant to the chlorine solution. But this is a challenge for the food industry. Because all the plants, there's outbreaks, you always heard about it. Today there is a 500,000 pounds of beef recalled in plants in Indiana because of E. coli L157H7. Why they started that? Most of the time because of the biofilm. Now we want to do the observation of the biofilm. We want to see what the biofilm looks like. We need a tool because we want to see the matrix. And lots of the time we can use electron microscope, but it's way expensive. So I give you example, at WVU's, only shared facility, which is a building behind us, at engineering building, they can use. And you have to go through a training process, get the certifications that you get. But how about the people just working in a general lab? That's why people create a tool, which is a laser confocal microscope. It's relatively less expensive compared to electron microscope, still expensive. Their major function is to see 3D images. And a good example is a bacteria biofilm. Okay, that's why the laser confocal microscope they are using. I have some research results I will share with you on Tuesday. We're going to go over it when the slides comes back. Okay, we get some time, so we're going to be very brief, talk about electron microscope. Electron microscope, we already mentioned that. You need to go through the training. I don't have that in my lab. I mean, we don't have that capacity to do it. So we have basically categorized electron microscope into two categories. Number one, we call SEM. What's that? Scanning electron microscope. Number two is TEM. What is that? Transmission electron microscope. So what's the difference? We already said the name is scanning. What's a scanning means? Backward and forth on the surface. So you can clearly see the surface. You also could see 3D images. But we only need to see the surface. So what we don't worry too much is a magnification. So scanning electron microscope, the magnification most of the time is relatively smaller, around 7,000. Much better than our light field microscope. Our light field microscope is maximum is 1,000. Okay, what is transmission electron microscope? We can see internal structure. And the magnification is very high, up to 20,000. Now, when we prepare the sample for electron microscope, it's very special. It's not like you just grab the bacteria and do the observation. They need a training. So basically what we need to do is freeze drying. And then we have to slice it. The freeze drying is usually using liquid nitrogen. Then we cut into very tiny slice. And when you cut in a very tiny slice in this area, we want to see the internal structure, for example. We call it a shadow. And this shadow, we have to emphasize it, to magnify it. You have to coat it. So we do a coating with silver nitride. Or sometimes using a heavy metal. We have to coat it. And then we underneath the microscope, we can do the observation. Now, if we want to go to see the major difference between electron microscope and the light field microscope, what is the major difference? Is the energy resource. Light field microscope is light is energy. Electron microscope is electron. So light field microscope, you see these things, basically is glass. How about the electron microscope? It is electron magnetics. That is a major thing. The second thing, because light field microscope, so the thing passing through the medium is light. And the electron microscope, the energy coming from electron beam. That's another major difference. This is make sure the subject is more focused. That's why it will be more magnified. And, but the equipment is huge. Well, I can bring a light field microscope just here, but I never can bring an electron microscope because it's a little different compared to all these things. Okay, let me just see. We have in your slides, you also could see the atomic force microscope. Atomic force microscope. This one. We don't have it. I worked with the project with my collaborator at the University of Connecticut. So this is coming from our local community's work. We will talk later on. There is a chemical product called semi-date 5.0. The major components is peroxyacetic acids and hydrogen peroxide, H2O2. You heard about that. At West Virginia, we have a lot of small farmers. The small farmers, they growing produce. You know about the FSMA, there's the Food Safety Modernization Act, which has come out is 2010 because of the outbreaks. So the small farmers, they are waived with the certification of the FSMA. So therefore, they need to do something to make sure their produce is microbial safe. So they're working with our small farm conference center to ask me to do something, to do the validation study, to see the peroxyacetic acid with hydrogen peroxide, how good to do the washing study for the produce. And we don't do one-step wash. We are using the triple wash, which is 3 steps, including like water rings, and finally we put in a tank, which is antimicrobial dipping. So we're using these antimicrobials. And once we use these antimicrobials, we want to see a little bit of mechanism. We find that it can kill bacteria, but how they kill bacteria? So we worked with our collaborator at the University of Connecticut, and we targeted the cell is Listeria monocytogenes. And you heard about that. The pathogen always in the outbreaks of the fresh produce, especially cantaloupes. So we did this using Listeria monocytogenes, which is also gram-positive bacterial cell, and we can see the cell after treated with peroxyacetic acids. We can see the width of the cell is changed, the height is changed, the roughness is changed. And this changing is more magnification or more significant with the increasing amount of the peroxyacetic acid, the hydrogen peroxide are using. So that's why I want to tell you one thing. From the clinical standpoint, why we need using this microscope? Because we need to do a rapid identification. So for example, people see the arms has a target eye. Oh, then the doctors know this guy has been to the ocean diving or like the marine activities all the time. So using dark field microscope, they will find Borrelia oh, possible Lyme disease. Then we use antimicrobials immediately to do the treatment. Now for us, when we are food microbiologists working for the local community, we want to try to see the mechanism a little bit, because then we will well understand how they kill bacteria. So that's why those like a laser confocal microscope, those atomic microscopes comes out, because basically those are research based. And some of the microscope is clinical based. But light microscope is more well established in about 100 years, so they're using the lab a lot. Okay, so we end up today, and the next week, Tuesday, we're going to talk about staining technology, especially we're going to talk about the gram stain, and also endosspore stain. We cannot do it, but gram stain we can do in our last class, which is the lab section. So you get familiar with how we do the gram stain. Okay? So thank you.